Good morning all. Myself Dr. Upender from Department of Computer Science, Akal University, Talwandi Sabo. First of all, I would like to welcome everyone who is a part of this webinar at this very moment. I hope everyone is doing well and staying strong as we move through this challenging time together. We have been going through a tough spot given the whole COVID situation. However, what we can't lose is the focus on what's important. For Akal University, that's always been a goal to educate the masses. This institute has been in operation since 2015, delivering their promise of quality education under the supervision of our Honorable Vice Chancellor, Dr. With the combined with the best efforts of the high qualified faculty. This institution has earned itself the reputation of a leader in the education. Under the supervision of our Honorable Sir, this institution has earned its reputation. Even during these tough times, we are under the COVID situation. We are doing our best to guide our students and provide them the opportunity to enhance their knowledge. Apart from the online classes, we have organized various online activities like webinar for the students to keep them motivated. Today's webinar would be an excellent example of demonstrating the same. Keeping our students' best interest in mind, the Department of Computer Science took this initiative to organize a solving biomedical problems through machine learning. And this webinar aims to provide the latest advances in the field of biomedical and health informatics, where informatics and the information technology intersect with the health and healthcare, life science, and biomedical. And the webinar vision is to provide the platform and exchange of valuable knowledge in the recent developments in the machine learning in biomedical and the health informatics. It covers the information of processing and analyzing of biomedical and health record using the machine learning, recent research tools working on the biomedical technologies. Our today's guest is Dr. Shandar Ahmed. He is from JNU, and I would like to welcome sir. And now I would like to brief introduction as he is a very renowned personality and he needs no introduction. Dr. Shandar Ahmed is currently working as a professor and scientist at the School of Computational and Interactive Sciences, JNU, New Delhi. He also heads the Skyway Lab organization, which is involved in an integrated approach to study life science and health related problems. He is a very enthusiastic scientist. He also ranked 4 on the list of best bioinformatics in India. Shandar Ahmed sir is a computational biologist interested in the role of genomic DNA shaped in transcription factor target interacting, and he investigates this using the machine learning and deep learning techniques. His works on predicting DNA binding proteins, genomic DNA conformational ensemble dictionaries, interactive functional annotation employing the transcriptomatic sequence and structure of based signature and clustering patterns of conserved residuals in transcription factors are some of the techniques he developed. He is also appointed as the research scientist at the National Institute of Biomedical Innovation and has won many awards. He published more than 100 research articles in very high impact reputational international journals. His area of interest are bioinformatics, big data analytics, and the complex systems. Now I would like to request our respected sir, Dr. Sukhjeet Singh, Dean from Akal University, to formally address our guest and say a few words. Please, sir.
Honorable Professor Ahmed, Dean of Computational and Integrative Science, Honorary University, Dr. Upender Kaur, Head of Department of Computer Science and Engineering, Dr. Praveen Kumar, faculty in the same department, dear students and other participants attending this webinar. It is my profound privilege to welcome all of you in today's webinar, which is based on the solving biomedical problems through the machine intelligence. As I remember, this is the 4th course curriculum enrichment activity of this semester being organized by this Department of Computer Science and Engineering. And in that sense, I would like to thank Dr. Kaur and Dr. Praveen Kumar further efforts in exposing our students to the renowned academicians and scientists of India. I'm not a person from this computer background or don't know too much about the machine learning. But few months back, when Dr. Praveen Kumar joined us, and after a meeting under the chairmanship of our Honorable Vice Chancellor, sir, he was participating on behalf of Computer Science and Engineering Department, and he approached Honorable Sir to initiate the interdisciplinary research activity using machine learning. And then I remember Honorable Vice Chancellor sir constituted a committee, and I was a member of that. And under that committee, I attended a series of meetings with the Economics Department and one with Commerce Department. And during that meetings, I learned it is a very, very powerful tool. This machine learning is a very powerful tool. It can be used in many research areas. From that day, I had a little feeling about this machine learning activities. Similarly, another chance I got to learn something about this machine learning. A few days back, I got an article which was based on the prediction of disease identification, behavior modification, clinical trial of such activity based on the machine learning. So in that sense, I was thinking this activity, which is based on the predictions of the on the various things using machine learning, it is a very useful activity. So I am sure this today's webinar, which is based on the biomedical solution of biomedical problem using machine learning, is going to be very successful event, and I hope students of this computer science and engineering they will learn a lot from the Honorable Sir. Before I say thanks to all, I would like to request Dr. Aman, Professor Aman, to visit us whenever he is comfortable, whenever he is free. Please spare some time to visit us and guide us so that we could strengthen our machine learning research activity here at the College University.
Thank you.
So I would not like to take too much time. Now I request Dr. Pinder Kaur to invite Sir so that we could start our students activity. Thank you very much.
Thank you so much, sir. Dr. Sukhjit sir, thank you so much for your kind words. We are really grateful to your efforts for organizing our webinar and guiding us in making it beneficial for a number of peoples. Now, I also like to welcome and express my sincere appreciation to the Dr. Shandar Ahmed who joined us today despite their busy schedule. And now I welcome sir, and please start, sir. And over to Shandar, sir.
Okay, thank you very much. And a very good morning. It's a cold day. And I hope we can all warm up with the little learning experience, which sometimes energizes our blood. And thank you very much for introducing me, and also mentioning, which is something not true, that my name is among the 4 top bioinformaticians. I'll correct this a little bit. So I would come 4th among the top bioinformaticians whenever 4 random bioinformaticians are selected from the country. So I have that kind of a feeling about myself. I am a student of this subject, and we all learn together. So there's no ranking as such that can put us in any hierarchy. And my lab is known as Sci-Fi Lab. I'll come to this. It's a very good occasion to meet all of you. And I was not aware about a very nice university, Akal University, coming up in our neighborhood. And definitely I would like to interact with the people here, students and faculty, in the future. So I start my talk with this. Let me share my screen. First of all, let me just check my voice is clear and audible enough. Volume is not any
Yes,
problem.
sir. Yes, sir. Audible, sir.
Yes, sir, you're audible.
Okay, so as you see on my screen, this is the logo of my lab. This is not a separate organization. This is just a research lab that I name as Sci-Fi Lab. And the etymology is that this comes from science and Y for query. So we do a scientific query, and as you see, Sci and Y combining to Greek and English alphabet. And on top, you see some Japanese character. This Sci-Y in Japanese means happiness. So this coincidence of these sounds allowed us to integrate it. Our school name is School of Computational and Integrative Sciences. So we tried to integrate whatever was possible, whatever was at our disposal. And Yesterday, I was giving a talk at in bioinformatics and biodiversity conference in Indonesia, and there I proposed something provocative, and I said science has always been data driven, but I specifically mentioned. Oh, I think I don't have that slide. Just don't. I think I opened the wrong slides. Give me a couple of minutes, please. Sorry. Okay. Okay, there you go. So I'm sorry about this. So I start with a little poem, which is about data science. So data science and machine intelligence come hand in hand today. And almost pretty much every problem of science and in social sciences and all other humanities also, machine learning and data sciences are entering in a big way. So I start with a little poem which I wrote in this, just to put this in context, how physics, mathematics, and other subjects stand in comparison to data science today. So this is with a little apology, of course, because it takes a little exaggerated view of the situation and little just kind for fun. So physics is fundamentalist and fussy. Why it is fundamentalist is because physics always looks for fundamental laws of nature, the basic laws of nature. So that is why it can be called fundamentalist, limiting itself to the fundamentals. And fussy. Mathematics is dumb and not yet easy, but because mathematics is very accurate, it tries to kind of model, identify the models and try to do things in analytical manner and try to know how the things are related to each other. Astrology, on the other hand, can draw conclusions too fast. From a very small set of observations, it can arrive at a conclusion. So astrology is hasty and crazy. Human brain, when we take our decisions in our life, we don't use mathematics, we don't use physics, we don't use astrology. Our brain takes decisions. We quickly, when we are lifting a stone, when we are lifting some empty glass of water or a glass with water filled in it, we have in our mind some calculations done. We decide how much force I need to use for my hand and pick it up. So our natural intelligence does not use physics. We are not calculating the Newton's laws of gravitation. We are not applying them. But our mind has already learned how much force is required to do a particular task. how much speed I should have to reach a particular destination in a given amount of time. So we do all those things which typically on the academic scale are done by physics, mathematics, and sometimes predictions, but we do this in our mind without actually doing all those conscious calculations. So our mind must be representing and doing these calculations in a different way. That's why the second stanza of this poem says, human brain uses none. Because for deep inside its terrain, the terrain of our mind, it knows to represent, test, and train without bothering to explain. It doesn't create the formal laws in our brain. Our brain can solve very complex problems without having ever studied mathematics. So data science rules this world today. For even if a solution is transient and predictions are noisy and incoherent sometimes, they work the best for most because they are intelligent. So intelligence works. Natural intelligence or artificial intelligence, intelligence works. We have to distinguish between what is natural intelligence and what is artificial intelligence, as we call it today. So I was talking about early on here that yesterday I compared these two great scientists in the field of biology who have worked on genetics and evolution. Charles Darwin, and they were almost contemporaries, you see, as you would see that both of them lived around the same time. And actually they passed away around within the period less than a gap of 2 years. So Darwin was looking at fossil data, whereas Gregory Mendel, who is the father of genetics, and Charles Darwin is, of course, the father of theory of evolution. So Darwin worked on data, and Mendel was working on the inheritance and trying to use a lot of mathematics, and also doing experiments in the lab on plants. Then, why say that Darwin was the first data scientist? Because Darwin never went to the lab to perform his experiments. What he did was he collected fossil data. And using the fossil data that he had, he modeled this data and theorized evolution, that is how perhaps that origin of a species has occurred. And that is how he explained the origin of species. So can we say that Darwin was the first data scientist? And not just Darwin, actually, if you think about other fields of knowledge, now I'm told that there are participants in this program from other fields. So if I take examples, say, for physics, so the laws of gravitation, and they were based on accurate collection of data on Kepler's observations of planetary motion. So Kepler's laws came, and the Newton's laws of gravitation came thereafter. So this is based on accurate collection of data and modeling of the data using some kind of mathematical formulations. And if I take another example for chemistry, the entire quantum mechanics was founded on the Bohr's postulates. So Bohr's postulates came from the careful observation of the hydrogen atom spectrum data. So the Lyman-Balmer-Paschen series, and then they was modeled that, okay, this is the data. This data can be explained if I relate these wavelengths to 1 upon n1 square minus 1 upon n2 square. So this mathematical formula was first devised, which was purely a data-driven formula. And this data-driven formula gave rise to an entire new field of study. And now this is well-established quantum mechanics. And there may be so many other examples where the data actually was modeled with the help of simple mathematics, sometimes complex mathematics and statistics, and led to a discovery. But today we talk about machine intelligence in a different way, and we talk about data science in different ways. So what is different today? So the big difference between the classical way of approaching data and making discovery from data and the modern day approach to making these discoveries is that machine learning can convert our data into models with arbitrary complexity without actually knowing what these laws are. So as this legend shows that you have these data, and this data passes through basic laws, and then you use or abuse this law. This is the traditional approach of discovery, the first horizontal line. And the modern data science goes through, completely bypasses the basic laws. It can actually take the data in and produce the answers to our questions without having to bother so much about the laws. So in some sense, people have in the past criticized this to be a black box approach, that I have certain gene expression values and I want to predict what will be the phenotype. So this gene, the features, thousands of features on the one hand, and a few classes of the predictions that I want to make. These predictions come accurate, but why is it that this particular transcriptome is giving this output and this particular transcriptome is giving another output? So this kind of the complete absence of this detail, what has given us the answer, and focus on the correctness of the answer and the validity of answer throws up new challenges. Because if you know the mechanism, if you know the basic laws which are giving rise to to an answer, we can possibly trust them more. But if we do not know what is giving rise to my answer, I have to find additional ways to validate, to understand what is, whether I should trust this outcome or not. Because machine learning often makes some predictions, and predictions are also made by machine intelligence and also by natural intelligence, and sometimes by complete natural stupidity. Sometimes people make absurd predictions, and there are people who would like to follow those predictions. So how can we distinguish between absurd predictions from the real, meaningful, and trustworthy, reliable predictions? That is the new challenge that is thrown when we try to model our systems in which the structure of the model is not very clear to us. So that is why the entire field of data science has emerged, where we have to not only understand, not only we have to model our data, but also have to assess the reliability of our models. How the techniques like cross-validation, leave one out, and other kind of the generalization value, overfitting ideas, overfitting, generalization, penalty for additional features. So all these techniques have additional. Yes. Can you hear me? Is that a message for me?
No, sir. Sir, I'm very sorry. I think there's someone is speaking over phone.
Okay. Okay, so I can carry on. So what I was saying is that the field of data science has emerged because we have become addicted to creating laws, with creating the models and creating the outcomes or producing outcomes and making predictions in the absence of a thorough understanding of the models that are giving rise to them. So this is one very important aspect of the data science. So machine learning or machine intelligence is not just about modeling. It is also about assessing the reliability and validity of our models. For instance, if I give you a rough example, that if I collect the X-rays from my fingers and I have a lot of data and I want to predict the state of my mind, I want to predict the neurological disease or something totally unrelated. So if I simply apply and without understanding the domain of the subject, without understanding the whole data that I'm analyzing, it is quite possible that I can create a very accurate models that I take the X-rays of my fingers and I can predict the state of my mind. But this is completely invalid or not correct. So how can we know beforehand that the model I'm creating are really valid? So this is one very important question that I will touch upon during this discussion, and also how the field has evolved and how machine learning, how we have moved from the classical approach of discovery to the modern approach of not so much into discovery of methods, but discovery of the answers. Why it is important is we have to, if you say that we do not know what the model is working and we still want to make predictions, why to do them? Just discard them. No, we can't afford to do this because we have so much of data and this data has so much of information. I want immediate answers to my questions. I have a lot of profiling of individual patients and I know that this person responds to a drug and this group of people do not respond to a drug. And I do not know the mechanism how this drug is, what this drug is doing inside my body. So I collect a lot of data. I collect transcriptome, my metabolome. I collect the genome of the person, etc. All the chemistry of the drug, the number of bonds, hydrogen bonds, the size of the molecule, etc. So I can collect thousands of features, and I have one set of data which says that this drug works. Another set of data that says it doesn't work under those conditions. So I want to have an answer without a mathematical, I cannot wait for the mathematical model or understanding for the mechanism. I want immediately to find a solution so that I can tell beforehand that this patient should be given this drug. So this is the problem. Briefly, I'm explaining to what is called precision medicine, that I collect the data about the drug, I collect the data about the individual, and I have the history of how this drug was administered and how different people with different profiles have responded to it. So using this, I can create a machine learning model. I may not understand what all this drug is doing to individuals, but I have the data which tells me that this is potentially, if you give this drug to this person, that this will work, or to this person it will not work. So these predictive models work like a black box. I will not understand what is going on inside, but the data science and machine intelligence can tell us the answer. So But then this model that I'm creating completely in a black box, whose mechanism I don't understand, requires a specialized understanding of what my model have done. Whether I can trust my model is making a prediction, but I might end up making the situation worse. I might be giving the drug to the wrong people. So can I really trust the answers that my machine is giving? Can I really trust this intelligence? That is a very important part of the machine learning process. So the creating models, now you have software, you have data inputs, you have data outputs, but then trying to understand what is going on is very important. But what is going on cannot be understood in a traditional sense as has been done traditionally in the past way about discoveries, but we have to adopt the new techniques for them. So in this science context, we can say the basic scientific discovery has seen three paradigms. Of course, there are other books recently who have written 4 paradigms over different paradigms they have mentioned, but I summarize them in 3 paradigms, easy to understand in this situation. So in the classical context examples that I gave you, the data collected by Darwin, the postulates given by Bohr, the gravitation laws by Newton, or any such examples, or Ramachandran plots. So these are the different examples where all that people have done is they have collected the data and they have used basic statistics, and from the basic statistics or basic curve fitting, they have tried to find an answer. So let me say this is an approach which simple statistics can, or simple mathematics can answer my questions. And I can also understand why it is giving an answer that I have. So simple statistics can yield the powerful results from a high quality data. Now here, why is it that the data is available to millions of people, the entire human population, but only some people make the discovery? Or why is it that discovery is not fast enough? So the basic difference between a good the people who can make this discovery and people who cannot make this discovery lies not in the mathematical power, but lies in the quality of data. As it is often said as a cliche that garbage in, garbage out, garbage out. If you put garbage data into a model, the model itself becomes a garbage. So the quality of data becomes more important and the fundamental thing for making discoveries from simple mathematics and simple statistics. So if just to give you an example, if the gravitational laws could not have been discovered if accurate measurements of the planetary motion were not available, planetary locations were not available. So when people record it accurately, okay, at this time the planets are here, at this time planets are here, and that is why we can conclude that this planet is going in an elliptical orbit, and this orbit is this speed of this planet in this location is this much, and in this location is this much. So accurate collection of data becomes very important. If you have accurate, high quality data collection and many other aspects, it is not just about the accuracy that you collect, it is also about many other aspects of data, such as pre-processing, like you may have redundant data, you may have biased data. So the data quality comes with a lot of questions that data scientists have to understand whether my data is accurate, whether what is recorded is correct or not. We never have accurate data. Most people who work with data science know that it will be impossible to have 100% accurate data. So what is the quality of my data? How accurate it is? What is the degree of the noise in my data? And what are the biases in my data? I may be collecting the data from only one set of population instead of an entire population. So these are simple statistical questions, how to do good sampling. So these are, first is the collection of data may have problems. Second is the data that we are trying to analyze may have been sampled from a large population and therefore the sampling may not be correct. So there are many issues, how to get a good sample and how to get a good data. But then once you have a high quality data, you can do basic statistics and basic mathematics and find some answers. So this is an example in 2004, long ago, when I was studying the protein structures and I wanted to predict. I had a set of proteins which bind to DNA and I had another set of proteins which do not bind to DNA. So can I just look at their structures and can make a predictor and can tell which proteins will bind to DNA or not, so that I can identify new DNA binding proteins. So people were doing very sophisticated and very complex mathematics and chemistry and biophysics at that time, trying to look at the atomic interactions, trying to look at the charge patches and all. So we in 2004 did a very simple approach that we just take the entire protein And we know that out of the 20 amino acid residue that protein is composed of, 2 of them are positively charged and 2 of them are negatively charged. So I simply count the total number of positive charge and negative charge. And with the help of the locations in the protein structure at some specific locations, I assign that charge in a very coarse-grained manner. And we can say that, okay, I can calculate total charge and I can calculate the total dipole moment and the quadrupole moment. Of course, they are not accurate. They are very rough estimates of the dipole and quadrupole moment particularly. But these 3 descriptors, because the quality of data, the kind of protein structures that we collected of a very high quality and the redundancy was well eliminated, we could identify as many as 90% DNA binding proteins correctly. So this is, again, the power of research, the energy of the research in this case has to be spent in compiling and curating a high quality data. Anybody can calculate this dipole moment in a very rough estimate, but they will not be able to make discoveries if the data quality is not good. So that is what I share with you, the experience of doing this discovery. And later on in 2014, we collected another high quality data where we looked at the protein's structures. When they bind to DNA, for instance, they go to genome. For those who don't understand biology, the protein transcription factors are some proteins that go to the genome and they find a specific location on the genome. And on these locations, they bind. And as a result, they start expressing or making proteins in the genes which are located nearby to that genomic location. Now, we try to understand where this protein will bind in the genome. Therefore, they want to know the structure of the protein. And these structures are solved with X-ray and others. So they would like to also know whether this protein will bind to this location or bind to another location. Because the location at which this protein binds determines what genes will express, what proteins will form, and what function a living organism will do as a result. So we were looking at this and we found that the protein structures, even if we model protein structures or we solve the protein structures, the structures before they bind to DNA and the structures after they bind DNA are different. It is like if you take an analogy of an individual's behavior, and if you want to know whether a person is very well-behaved person or a very rude person, how would you know? You cannot monitor that person for a long time. But then that person, whether the person may be very well-behaved in a company of X number of people and very rude in the company of Y people. So it is that type of a conformational change that the protein undergoes and can be predict what changes will take place if the protein binds to DNA. So we collected again the entire set of protein DNA complex structures from the protein data bank, and we divided the kind of conformational change that these proteins undergo can be grouped into 6 categories. And then each of these category has a different implication. Some of the changes make the protein DNA complex strong. Some of them make them very specific, they will bind only specific locations, etc. So the specificity and stability, as you call them, are the properties that can be determined from these conformational changes. So this helped us to understand a lot of protein DNA interaction molecular mechanisms. So these are, again, what we were doing is very simple statistics. We are collecting high quality data, looking at, okay, this difference and this difference. We take a simple t-test, we take a difference in the mean values, and we can say, okay, this category of the conformational changes is different from this category, and therefore this should be the behavior. So these simple statistical principles could lead to interesting results, which are very well corroborated by experiments. But then everything in nature is not simple. So because as I write here, simple may be also simplistic. So we go to the next stage when why we say simple is not sufficient, because if I study 2 behaviors of X and Y, and I put X and Y together, they behave differently. So when the 2 individual features in the computational science language, or 2 different properties of the same system come together, they interact with each other. The features interact with each other, not just the systems. So the properties of the behavior that we are studying, for instance, if we are looking at the 2 proteins, so 1 protein and 2 proteins are, when they are interacting with DNA or performing any function in isolation, and when they are coming together to perform another function, so AB, if one is A and another B, the AB is different from A plus B. So this is a complex behavior. This cannot be modeled from simple statistical models such as Bayesian statistics or something, unless you incorporate additional parameters there, which specifically define what kind of interactions you are trying to look for. So the simple models of statistics, such as multiple linear regression or the naive Bayesian classifiers, will not work whenever features are interacting with one another. So now the solution, if you still want to stick to the statistical models, what will you do is you will engineer new features. If I know that there is a feature X1 and there is another feature X2, and these X1 and X2 are interacting and to produce something Y, then I can create my model whose inputs are X1, X2, and X1 into X2. So X1 into X2 will model the interaction between X1 and X2 features. So but then I try to model this. If I have just 2 dimensions, my number of dimensions become 3. But if I'm working with say 100 dimensions and I have to add all the pairs of interactions, my numbers start increasing very fast. And if I have to introduce 3 feature interactions, then number will go very high. So the actual number of parameters that you have to train in these systems will become very large. And therefore, the traditional, or we can call them naive models will not work very well. Even 1 dependence or 2 dependence will make our models very high dimensional. And these very high dimensional models will easily produce very good outcomes and very good accuracies on the data on which they are trained. But they will produce no value on the data which the models have not seen during training. So this is the problem, that the problem of overfitting is what is at the heart of good machine learning models, that our models should not only work on the data on which they are developed, our models should work on the data which models have never seen before. So for this purpose, we often do what we say, we keep some data aside, we finish our modeling and test it once. Now, what many of times people do is, when they are trying to publish a paper, when they're trying to develop a model, they keep a data aside, they develop a model on a training data, they check whether it is working on the data that they have kept aside or not. Then they say, okay, this is not working. Then they try another model, they again train, and then they again test. They keep on doing it. And then pick up the model which worked well on the left alone data. So in this process, they are actually training on the data, because they are still testing their model on multiple times on the same data, which they had left out. So it is not really a left out data. It has been included in the model in an indirect way. So ideally, I should continue to develop my model, validate my model until it has been completely tested and finalized. Once I have finalized the model, only once I should use the left out data and test it on it. And that should give me the model performance. It may be a disappointing performance. My model may not be working at all. So then we have to become experts, so that when we are training on our data, we know what to expect on a new data. If we do not have to use a left out data, we simply keep on trying on the data that we are training on our model on, and we can also assess at the same time what is the expected level of generalization on the data that has not been used during training. So this is what where we try. So if the statistical models, naive models, try to generate a very high dimensional model data, this high dimensional data will produce highly accurate models on your training data, which have no value for the real application, because they have no generalization possible, because the models are what we call overfitted. So machine learning helps in this case, because the machine learning does not utilize does not rely on explicit description of the relationship between complex features and the outcomes. What it tries to do, for instance, a neural network, if you have, say, 1,000 input features and you have a single outcome of binary outcome, plus or n minus, so you can simply have 2 nodes in a hidden layer, and with the help of just 2 nodes, by increasing the parameters very little, you can create a very complex decision surface. So this kind of a complex surface cannot be created by, say, for instance, defining a polynomial or defining a multiple linear regression with engineered features which have the interacting pairs included in that. So that is where the power of machine learning has become possible. Particularly the neural networks have become very powerful tool in this case. So here a biological example that we did, again, we have been working on these systems since 2004 onwards, and we continue to make advances. So one problem we look at that there is a DNA, there is a protein sequence, and only some of these parts of the sequence, the residues, actually interact with DNA. So we can predict by looking at the sequence what are the residues that interact with DNA. The first level of information comes from the fact that DNA has a negatively charged backbone, and therefore any part of the protein sequence which has positive charge is likely to be interacting with DNA. So we look for the positions of arginine and lysine, and we possibly say that if there is an arginine here, then it is possibly interacting with DNA. But then the arginine, which is surrounded by certain residues, may not interact. So the surrounding residue information becomes important. But the surrounding residue behavior is not additive to the target residue that we are studying. So we have to model some complex behavior here, that if this is my position X, and I want to know whether this will bind to DNA or not, and these are my surrounding residues, then I have to model that, and then model I can do with the help of some encoding, and we can use a complex neural network, and I can have a model with a small dimensionality and with a relatively few number parameters and try to predict. So this kind of a non-additive behavior or the cooperative behavior between features can be modeled with the help of machine learning, in this case, neural networks. Another study that I would like to cite here is that the looking of the clusters of conserved residues. So conserved residues are those that in the protein sequences, there are some positions in the protein sequences which do not change as the proteins evolve from one generation to other or from one species to another. So they do not change. If something is not changing, it must be important for some function, and that's why this nature is not allowing these positions to change. And we studied comprehensively a large set of such conserved positions, and we found that these unchanged positions occur as clusters. So that is how we defined clusters of conserved residues. So these clusters of conserved residues had the information about their function, what kind of functions these proteins or these regions perform. And what we found that the unchanged positions occur as clusters. They do not act individually or as alone. They work in groups. And if I, for example, if I have a protein structure, I remove one part and I check the difference. I remove the second part, I check the difference. Suppose difference in first case was X1, difference in the second case was X2, and I remove both of them at the same time, then difference is not X1 plus X2. The difference is either less than X1 plus X2 or more than X1 plus X2. That means the role that X1 was playing, the first residue was playing in that location, was dependent on what is there at the position X2. So these kind of dependencies cannot be modeled easily unless we know the explicit relationships. But machine learning can try to capture this data and try to identify these relationships in an implicit way, where you do not know how these residues are interacting, but can still answer what is the expected change in the free energy or whatever the outcome we are analyzing. Okay, so then I described the two paradigms. The first paradigm of the data-driven discovery was based on simple statistics and simple mathematics, basically what you used to call earlier curve fitting. So you have data and you want to find what is the function that fits very well here. If I have, say, I do the Cavendish experiment in physics and I collect a lot of data on the charge and the force between two charged objects. And if I find that all these data points are falling very well on the relationship of 1 upon r square, so this will immediately lead to the discovery of Coulomb's law. So because I have now Q1, Q2 upon r square, and Q1, Q2 I fix, and I keep computing the force between two charged particles over different distances. And using these observations on the XY plot and the distance versus the force that I observe, if the data is falling very well on the mathematical equation of 1 upon r square, I have made the discovery, okay? This is the law. So this is the classical approach of making data-driven discovery, fitting the appropriate mathematical and statistical functions onto our data. And machine learning goes one step beyond. It makes this process a little blind or black box kind, where the problem is defined. This is my data. This is my outcome. Just try to make a predictor so that this applies on new data. This is the way the problem is defined in this second paradigm of knowledge, where machine learning is the dominant force. Now we move one step further in the current paradigm. We are going even beyond that. We do not define these features so very accurately. We have data, say we may just have the natural language processing. You may have recorded my audio, and from this, you have a lot of audios, and then you have some people may have liked this or this lecture, some people may have not liked this lecture. And then you collect the data about those individuals and those speakers and the individuals who have liked or not liked, and try to combine these features and try to predict, okay, this is my audio. Can I predict this audio, this lecture will be liked by the users or not? So that I can get some kind of guidance for the future lectures. So here I have a more or less audio and video are much better defined, but it's a very complex. If I just want to feed the entire video, and it is say just even 10 minutes video, you have thousands of images, frames, and each frame is made up of a very large number of pixels. So if I want to present this, just I want to create a black box that can predict whether this lecture will be liked or not. Some people have worked, for example, to look at a movie, and from the movie they want to predict whether a movie will become a hit or a flop. So this may be far-fetched at this point of time, but still people are trying, and this is but something short of that is already possible. That we collect unexpected information about our systems, and we try to make predictions about them. So what are the challenges in this situation? And these challenges are different from what we were facing in the classical machine learning regime. So here, the challenge is that the amount of data that you have, the amount of labeled data, as you call it. So labeled data means I have, say, 1,000 images, and I know about 500 of them are type 1, 500 of them are type 2. I have cancer cell images, and I know these cells are cancerous, these cells are not cancerous. I want to develop a classifier on this. Now, I have each of these images is very big, and I want to collect information about them. So what do I do about this? So now I am in a situation when the dimensionality again is huge and the data is small. So conservative approach of modeling our data has been that my data objects, the size of my data objects should be higher than the dimensionality, my dimensionality. We know for always that if we want to fit a unique straight line in a three-dimensional space, I need at least 3 points. If I have only 2 points, I can fit infinite number of lines. And similarly, if I have less data and more dimensionality, then I can have infinite number of models that can fit my data with 100% accuracy. Which of them is correct? We do not know. So this is the problem of this is the problem that we have to face when we're dealing with large amounts of data. So I talked about very high dimensionality of the data. Second problem is that my data may be natural language processing, where the data is not very well structured. So which is often the term big data term is used for that. So how to represent this data, how to exchange this data, and how to model this data, that becomes the problem. How to even scale the data. Some data is collected at a different scale, another data is collected on a different scale. Gene expression, somebody has collected from RNA-seq, somebody has collected from RT-PCR, somebody has collected from microarray, and they are on a different scale, they have different variances, they have different mean values. So how to put them together? The challenges of unified normalization, the pre-processing challenges become very important. So deep learning is the actual model, but even pre-processing, most of the data scientists spend a lot of time on pre-processing the data. And by one estimate, 70 to 80% of data scientist time goes in pre-processing, not actual modeling, because for modeling, they have software, they have, okay, once their data is ready for model, then they can simply use an existing code and run it. So these are the challenges we have. I show you some of the problems that we have addressed, and that will give you an, how much time I have now?
So you can continue if you want, like for another 10 minutes.
Okay. So one problem, as I've been talking about, is the protein DNA recognition, and there are a number of questions in protein DNA intelligence. And one problem I quickly pass through here is because there will be more people not from structural biology or biology. So I'll come to problems of more general nature also in this discussion. But one question that we are trying to address is to look at DNA structures. So I introduce this from here first. Yeah. So one transcription factor has many target positions where this transcription factor or protein can bind in genome. But in reality, in the cellular state, transcription factor does not bind to all those locations. We know that a transcription factor has a motif, and this motif is just 6 base long. And the 6 base long motif is located at thousands and sometimes millions of locations in the genome. How is it that transcription factor selects some positions and does not select some of other positions? So this Venn diagram shows that the data collected from the same transcription factor known as STAT3 from 4 different cellular conditions, only 35 sites were common to all of them. Whereas 1,547 sites, which is shown on top, were unique only to macrophages, and the shown on the bottom is embryonic stem cell 2,148 sites, which are bound by STAT3 only in embryonic stem cells. So the point here is that the same transcription factor, when it goes to different cells, performs totally different functions. It regulates different genes and it binds to different locations of the genome. So how is it that the genome in one cell is different from genome in another cell? So what is going on here? This is a big enigma that people have been trying to solve, and we have contributed to this field. One of the roles is that of cofactors. What are the other partners available in different cells? So if I have X partner available, I will bind to X locations. If I have Y partner available in another cell, other genes, so I'll tie up together and I'll bind to another locations. So it is our social behaviors or our social preferences are also determined about who are our friends and who are the friends available in the society for larger interaction. So this is intuitive, but we looked at this is not sufficient. The cofactor behavior is not sufficient. There is something intrinsic to the genome, and that changes across the cells. So this is just this same information presented in a different way. So we try to look at whether the location at which the transcription factor is binding, whether just that location is important, or whether the regions surrounding that, where the transcription factor is not actually binding, are also important. So using a purely data-driven approach and using the simple machine learning approaches, we were able to identify that most of the information is contained in the target where the transcription factor binds. But then the genomic locations are then around that are divided into 4 parts, about 50 base each. In the first 50 base, you have what we thought is probably the stabilizing region that provides the presents the site to the transcription factor. And then there is a steering region, because then the transcription factor may be looking for the site where it has to bind. It cannot find the right sites. It reaches some wrong site. So at the wrong sites, that something is happening there in the genome, it tells, okay, don't bind here, just rush away from here. So something that throws it away quickly so that the transcription factor does not have to waste its time. So this allows very fast reach of the transcription factor to the target sites, and also provides, and this behavior of the surrounding region is also specific to the biological conditions. This is what we were able to model with the help of creating the dictionaries that we collected the entire genome, and so we can map the molecular dynamic signatures to the entire genomes, and we could model the behavior of entire genomes in this way. And we explained the behavior of thousands of transcription factors and grouped them into 4 categories. And we found that there are some group of transcription factors where only the target position is important. There are others for which the target is important and surrounding regions are gradually less important, whereas some have modular regions, and in some cases, target is not important at all. They are like people, okay, where I want to go, I have a destination of my own. Wherever my friends go, I will go with them. So this is this 4th cluster of transcription factors. Here, the transcription factors do not have any preference for their targets. They bind to locations wherever their cofactors take them. So these are the discoveries made possible through rigorous modeling of neural networks, and then lasso, and a very huge data has to be modeled in this, because each transcription factor produces millions of sites, and the data that we have to model here is about 40 gigabyte. So about 40 gigabyte data has to be modeled and trained, and then these outcomes came from that. And clustering of a large data is 40, and you have million versus million kind of comparison, and it's a huge computationally demanding exercise. that we successfully completed. So I'll skip this particular project that we are doing. I'll also touch upon briefly some deep learning models. So again, I have already discussed with you the dimensionality issue, and here is some putting it in perspective. What is the kind of dimensionality we are talking about? So if you're looking at the MRI images from brain, you have, say, a single image has 600 into 300 pixels, which is upon 180,000 features. And if you are feeding the entire image as the input to our model, we have huge number of features. So the classical approach to solve this problem was to not use the entire feature, but try to create features from them, feature engineering. So patch size, so that the entire, instead of pixels, we find out the lesion size. So you have one feature. You take this lesion geometry, topology, depth, etc., or so other properties. And so you can have just a few features and you can model them. But the modern deep learning approach is to use the entire image as the input, the entire pixel by pixel representation as the input. We all study this ECG diagnostics, and here if we take a 1 hour recording, you will have 3,600 per hour record in an ECG that we have to model, and we have to try to see whether in some locations you have erythemia. If you want to create a machine, then can predict, okay, some anomaly is happening to this patient and then they will need immediate attention. Suppose you are in a hospital and you want to create an alarm system, and the doctor should be called only when certain anomalies happen in ECG. So this kind of automated diagnostics, real-time diagnostics require, again, another high-dimensional modeling. And this is another example of gene expression or co-expression where these dimensional, they can be 10 power 8 if we are looking at co-expression. And in all these problems, the amount of data that you have is much smaller than the number of dimensions that we are trying to model. So the question I already discussed, what is happening if our dimensionality is too high, it leads to overfitting and creates many alternative hypotheses. So this is equivalent to what I said. If you if you have just 2 data points and you are asked to fit a 3-dimensional in a 3-dimensional space, if you just have 2 data points, then you can create infinite number of models that can completely fit these 2 points. So there can be many, many models which are possible. We do not know which one is the correct model for our data. So we do a number of, so what happens, what allows deep learning to work in this case? The dimensionality of the deep learning models is very high, but still deep learning models are widely used. So we are in a regime where we have even done away with the data size requirements. And we are now working with, so these are some of the publications that we have. So the point here is that in the case of convolution, so these are the typical model of a convolution, this one. So you have all the features that we are trying to train go into the model, but then we do something to our model to reduce to create an internal representation of our dimensionality by way of creating convolutions. So these convolutional representations actually do not directly model the images pixel by pixel or the features point by point. They try to first build a relationship between these features, and they are not learning on the target output in a supervised way. They are learning on the feature representation itself. And after this convolution, then you have some feature mapping and pooling and feature mapping, and then you create a predictor. So this kind of a fundamental principle of deep learning to be able to create some kind of convolutions and kind of relationships between features internally are at the heart of the power of deep learning models. There are others concepts like the transfer learning, when you train the model on one data set and you utilize it on another data set. It also helps in reducing the requirement of the data sets. So these are some of the ways in which deep learning outperforms the classical models of prediction. So I now conclude. I think I've exhausted my time. So data science has always been fundamental to scientific analysis and discovery. Field of data science and also machine learning has moved from simple statistical analysis to classical machine learning and now deep learning models. But one should not assume that deep model will work better than a shallow or classical statistical model. The number of factors contribute to which model is more suitable. And that includes data availability, system complexity, and we should not assume again that the most complex model is more accurate. In fact, one should go from the minimalist approach try to create simple model first and then go to the complex models and successively assess which model is performing and what to expect in terms of generalization, not in terms of accuracy. Finally, to conclude, I describe it in again a few lines. Mimicking brain, shallow and deep, machines learn while man is asleep. Without complex equations or laws, making use of errors and flaws, because you need to have error and you train the model backpropagation with the help of errors that come in the output of a neural model. So making use of errors and flaws, regressive, divisive, and classifying, they're all machine learning terms. Recursive, divisive, and classifying only in AI are so gratifying. So thank you very much. And this is my lab members. All of them are not represented here. The slide is slightly old. I have to update it. Thank you. I'll take questions.
Thank you, sir. It was really an enlightening session. We are grateful for your time and effort. You took to share thoughts and experience with us. I thank you bottom of my heart. If you people have any question, you can ask the question. So I'm sure many of you have some questions. So the floor is open for questions.
Sir, I have one question.
Yes.
As we are having first year students and they are beginners, and I want to know what are the prerequisites for a beginner to start a research with the bioinformatics streams.
So it depends on people are coming from the background of computer science or biology.
Computer science. Sir.
Second, computer science.
Computer science.
Yeah. So now some people do distinguish between computational biology and bioinformatics. So bioinformatics is more closer to people who computer science people who do biology. One good thing about bioinformatics is that this is a subject that you can study and you can start anywhere. You cannot start physics from anywhere. You cannot study quantum mechanics if you do not know equations and if you do not know the basic mathematics to deal with the wave functions, etc. In biology, you can focus on a specific problem and try to solve these problems without having to know the entire field of biology. So first thing is for the computer scientist is to identify the problem in which he or she wants to contribute. And try to look at what is the state of knowledge in this field. There are many problems that computer scientists are solving. Some of them I touched upon today, like image classification. There are many different aspects of image classification that people are still working on. For instance, cancer cell state, and different types of images. People are studying X-ray images. People are studying the MRI images, whole slide images, etc. Different types of images, and then trying to solve different problems. For instance, most recently trying to predict COVID from the X-rays of the lungs. Now, these are the problems for which data are available, and people are developing classifiers or predictors with different types of models. And in computer science, people are expected in this case not only to model an existing data, but to improve the performance of these models and to be innovative about methods part of it. If you just, as a computer scientist, one warning I would like to give you people is that as computer scientists, if you simply apply an existing software on some data picked up from the net, this is not sufficient. Because this job can be done by a biologist. You have to think in terms of how you can change or alter or improve the methodology of performing that task. And when you go to an interview, and when you, since many of you are students here, and it is right time to learn how to prepare yourself. So when you go to an interview, focus on this aspect of your work, that this is the method that you've improved. And just do not say that I have applied X, Y software and solved it and modeled this. Because this is the job a biologist can do very well. As computer scientist, you have to go beyond that. You have to identify this was the lacuna in the field, and this was something incorrect in the models. And so these examples could be, you identify some prediction was being done from images of one type. You say, okay, this I can now integrate another property, behavior of data, say transcriptome, and I can combine them, and I can answer this question more accurately. So this methodological advance has to be at the heart of your research in the future and your technology development. And a lot of data is available. You can take any data from the net. Of course, you must be aware of Kaggle and UCI. So take those data. Already a lot of codes are available for computer scientists to play with, and that is how they can learn. So that is where you can directly take their codes, but don't stop there. You have to think of how you have to understand these codes and try to engineer those codes to solve these problems in a better way. Sometimes defining new problems, and this has to be done in collaboration with a biologist, whether the problem that you're defining are really useful for the biological application or not.
Okay.
Really thankful, sir.
Okay, thank you, sir. So I'll have a very generic question. I'm not going to ask you a specific question. Like I have been asked many a time. Like, though I can see you have answered that question, this question in your slide, but if you can, generalize the answer, it will be great for us. What is difference between artificial intelligence, machine learning, and deep learning? This question people throw to me many a time, and believe me, I don't have a particular answer for this.
Okay, even I'm not an expert to answer this question, but I'll give it in the best possible wisdom that I have. So first of all, separating the deep learning from classical machine learning. So deep learning is the term that comes originally from the neural networks. Typical multi-layer perceptrons in the past have been just one hidden layer and one input layer and one output layer. These are the traditional multi-layer perceptron models that have been widely used for a very long time. And in fact, there was one theory that said that any data of any complexity can be modeled by a single hidden layer by just increasing the number of nodes in it. So one hidden layer is sufficient. That was the understanding. And but of course, how many nodes we need in that hidden layer, that matters. So that this remains a question. Our lab is also investigating this. I think Anima is attending this program and she's working on this. How long should be this hidden layer? And then when we say deep multi-layer network, in the deep network, we increase the number of layers. So we do not have one layer, you have multiple layers. So it becomes deep multiple layer, multi-layer perceptron or deep neural network. Now the question here is why deep models were not used earlier and now it has become popular. One big reason is practical. In fact, practical reason applies to neural network also. Neural networks were discovered much earlier, but they became popular much later. Because when they were discovered originally, the discovery of backpropagation method was also made, but we did not have enough computing power to solve real problems. When computing power improved, people started adopting and they found neural networks work excellently. So same is true for deep learning models. Deep learning models become very difficult to train because of the large number of layers, the backpropagation throws up challenges into gradient, many challenges, such as one common problem is the gradient vanishing problem, that the gradient becomes zero and therefore no errors cannot be backpropagated to the input layers. So there were many reasons where people were trying to use not deep, but what we can call shallow neural networks. So in that sense, this is deep. But then from the multi-layer network, then many different versions, deep versions of the classical neural networks have come. One of the most successful one particularly for image classification is CNN, convolutional neural network that I briefly touched upon. And there are a version of the recurrent neural network. Earlier we used to have recurrent neural network, and then you have a deep version, which is LST. So essentially, a deep model is one which uses multiple layers in a neural network, and it is frequently and perhaps predominantly used in the context of neural networks when you have multiple layers between the input and output features. Then they are deep. And this is between machine learning and deep learning. For the artificial intelligence is a broader theme where not only the machine learning appears, but also certain additional things such as IoT, how to collect this data. So AI may also include certain aspects of data collection, data sensors. And artificial intelligence is not just about machine learning theoretical models or computation models, but little beyond that, which may also include the hardware part, which may also include multiple decision-making components. So artificial intelligence in that sense is much broader than machine learning and deep learning.
Thank you, sir. I have one another question. It's again generic in nature, not very specific. Uh, like creating machine learning model is sometimes very challenging. And how to know which model we should I should select, like that. Obviously, that will depend on the data which you are taking. But nevertheless, if you can shed some light on
So
this.
our rule of thumb is that we should be minimalist in our choice of model. Minimalist means that we should not try to use a very complex model when our data is not really complex. So we should probably start going from simpler models and gradually increase the complexity of our model and see how they're performing. And every time we add new complexity to the model, there are ways to penalize for the complexity. The scoring functions. So for instance, traditionally we measure the model performance with the help of AUC or the accuracy. But there are also the classical concepts of the AIC, for example, where we penalize for every new feature that you add to the system, you penalize the accuracy. So this is models should be selected based on the, of course, if you already know what kind of relationship exists between your feature and your outcomes, so that will give you an idea about what models will be appropriate. If you know that you are dealing with a time series data, so you have some idea that perhaps LSTM will work better than people, but people have shown that CNN works there as well. So and for every time new model comes and a new problem comes, different techniques people keep on developing. But a broad principle would be that do not apply the most complex model from the beginning. Start looking at your data, visualize your data, what you call exploratory analysis. Visualization of data is very important. You should look at what is the relationship between features. Try to see the regression relationships between your features and between features versus your outcomes, etc. So visualize, understand, and explore your data. Modeling is the last step. And ultimately, you should spend a lot of time in exploring your data, investigating your data, understanding your data. Modeling will be easy part because you have, as I said earlier in my talk, that if you have the code and you know if your data should be represented in this way, then you can easily model this data. Spend a lot of time before going to the modeling, spend a lot of time in understanding your data, exploring the relationships between features and all into one-to-one basis if possible, or trying to do other analysis. For there are many approaches to do exploratory analysis. So spend a lot of time on exploratory analysis, reprocessing of data, bringing the data on the same scale, same orientation. If you have to image, perhaps align these images if your model requires to do that, etc.
Thank you, sir. If there's any other questions, you can ask. You can ask now. You can also write in chat box. So I'll wait for another maybe 10-20 seconds. So I guess there's no questions left. So thank you, sir, for this enlightening session. It was a privilege that you have given us a very enlightening session, and I hope you visit our campus someday. And if there's, ma'am, if you want to say anything to sir.
Sure,
Yes, sir.
maybe I'll just drive down. I have driven down to Jalandhar and other places, so maybe I love driving. So maybe one day I'll just stop.
Yes, yes, I know how much you love driving.
Yeah.
So
Thank you so much, sir, for such an informative session and on bioinformatics. And it is a great deal in science that we should learn and go research into this area. And it is a fascinating topic, and you wonderfully articulated this. And really thank you, to sir, for give us your valuable time. Thank you, sir.
Thank you very much, and you're all welcome to contact me. And of course, the faculty are more than welcome to contact me anytime if I can do something, and students as well. So feel free to interact with me and my lab members as well. That can be found on the net on my lab website. Thank you
yes,
very
sir.
much. Yeah.
Thank you, sir. Thank you very much.
Thank you, sir.
So you can leave, right? Like those who want